Okay, so as Melanie said, I will provide a brief overview about the virology and biology of SARS-CoV-2, or as can advance the slides. Which is short for severe acute respiratory syndrome coronavirus 2. And I will, in today's talk, just call it SARS-2. And as you're probably aware, it's the causative agent of COVID-19, the disease, which is a similar concept that we're already familiar with HIV, or human immunodeficiency virus, causing the AIDS disease. And as of this morning, there were over 3 million confirmed cases of SARS-2 in the world, with over 1 million, so a third of those cases in the United States. And so coronaviruses get their name from the appearance on electron micrographs. And so if you look in the right top corner here, you can see an electron micrograph of these virus particles. And surrounding these particles is the corona, or the crown. And this crown is driven by the spike protein. So if you go to the bottom, you can see a cartoon of these virus particles, and in pink is the spike protein, or the S, which is embedded into the envelope. And this, so these are envelope viruses that are roughly 120 nanometers in diameter. And in addition to the spike protein that protrudes from this envelope, there are also other structural proteins, the membrane, or M protein, and E, or envelope protein, that are embedded within this envelope. The fourth structural protein is N, or nucleic capsid, and that surrounds the viral genome, which is of positive sense single-stranded nature. And it's a roughly 30 kilobases in length. And coronaviruses in general, they are spread by the respiratory and or fecal-oral routes. And these viruses are divided into 4 different genera that you can see here: alpha, beta, gamma, and delta coronaviruses. And I have listed all of the human coronaviruses here, and those are found in alpha and beta coronaviruses. And the top 4 here are the common cold coronaviruses, and then the emerging coronaviruses, SARS-1, 2, and MERS, are found in beta coronavirus genus. And what we know about their ancestry is that gamma and delta coronaviruses have an avian ancestor, while the alpha and beta coronaviruses that emerged from a bat ancestor. And so to look at the genomic organization of SARS-2, you can see in this cartoon here that the virus expresses a great number of proteins, up to 28 proteins. And the non-structural proteins are expressed from the 5' end of the genome. Now, these are the proteins that encode the replicase enzyme. So the RNA-dependent RNA polymerase that replicates the genome, and other accessory proteins, as well as the proteases, which can clip the polyprotein into its individual pieces. The 3' end of the genome encodes the structural proteins, the 4 structural proteins that I've mentioned earlier, and up to 8 accessory proteins, which modulate the innate immune response and the host. And if you're interested to learn more about how these various proteins and mRNAs encoding them get expressed, there's a 1-hour YouTube video from from Britt Glanzinger that goes into more detail for those interested. Now, for today's talk, what I would like to do is focus on the steps in the viral life cycle. And the reason being is that each aspect of the virus life cycle represents an opportunity to disrupt the infection and thus is a target for antiviral drug development. And so we'll talk about virus entry, virus replication, and then assembly and release as we go through the slides. And so let's first focus on coronavirus entry. So as with every virus, the virus engages with a host receptor and then has to cross the limiting membrane of the cell and to deliver its genome into the cytoplasm. And so for coronaviruses, what we know is that there are 4 different receptors. So the SARS-1 and 2 as well as NL63 use ACE2, the angiotensin converting enzyme 2. And MERS uses the dipeptidyl peptidase 4. 229E uses aminopeptidase N, and then OC43 and HKU1 use a modified sialic acid to bind to the cell surface. And the protein that mediates this is the spike protein. And you can see a cartoon here of the domain structure of the spike protein with the N1 domain, the N-terminal domain being responsible for attachment. So there's it encodes the receptor binding domain, while the S2 domain has the transmembrane domain that anchors the spike in the viral envelope, and it contains the proteolytic cleavage sites, which are required for fusion of the viral envelope with the host cell membrane. And in a recent study that was just published in Cell, Hoffman et al., they demonstrated that ACE2 is the receptor for SARS-2. And you can see the data on the right side here. And what these authors did is that they used the pseudovirion assay. And so instead of using the entire SARS-2 virus, what they did is they took a VSV, a vesicular stomatitis virus, and then just replaced the G-glycoprotein with the spike proteins of these 4 coronaviruses. And then they ask whether BHK2 cells that express these different coronavirus receptors would be able to mediate entry of these pseudovirions. And what you can see is that both SARS-1 and 2 were able to use human and bat ACE2, but not the other 2 receptors, while, as expected, 229E used APN and MERS used the DPP4 receptor. And further confirming that ACE2 is involved, when you then use antibodies against ACE2, you can prevent infection of the pseudovirions, either from SARS-1 or 2, when you use increasing concentrations of these antibodies. And of course, receptor expression is often linked to tropism, which means which cell types are being infected by a given virus. And that was also investigated in this recent Cell paper. And so what you can see is VSVG pseudovariants at the top compared to SARS-1 in the middle and SARS-2 on the bottom. And what the authors did is they basically just asked which different cell lines could support pseudovariant entry. And you can see listed at the bottom here several different human respiratory cell lines, then a kidney cell line, a liver cell line, and a colon intestinal cell line. And so except for this one human respiratory cell line, all of the human cell lines were able to support virus entry, in addition to the vero cells, which are monkey cells, while mammals or bat cell lines that were less related to humans did not significantly support entry. And this actually lines up well with what we know about ACE2 expression. So this is just data from the Human Protein Atlas, where you can see that ACE2 protein expression is very high in the gastrointestinal tract and also found in the liver and the kidney, as well as some other tissues. And so, however, just binding to the receptor is not sufficient for the virus to enter the cells. And at the top here, this is a diagram of what coronavirus entry steps look like. And so once the receptor is bound by this the spike protein, in this case it's the trimer, and it's the S1 domain that binds to it, there needs to be a proteolytic cleavage event that occurs so that the S1 trimer is displaced and the fusion peptides are released so that they can do their function. And it's shown much better in the lower diagram, where you can see these orange rectangles, which represent the fusion peptides. And so once that cleavage event occurs, these fusion peptides now are free to insert into the host cell membrane. And these trimers then undergo conformational changes. They basically fold up on top of each other. And it's that that pulls both the host cell membrane and the viral membrane close together so that they can fuse. And so based on what we knew from SARS-1, the same authors from the Cell paper also looked at what are the proteases for SARS-2 that mediate this proteolytic activation event. And they tested two different proteases, TMPRSS2, which is a transmembrane protease, serine-2, that is inhibited by camostat, and then cathepsins, which are inhibited by E64D. And so you can see the data here. So vero cells express ACE2, but then they do not express TMPRSS2. And so in the cells when only cathepsins are active, if you inhibit the cathepsins with E64D, you can see that SARS-1 or SARS-2 entry is being inhibited, while the TMPRSS2 inhibitor did not have any effect. However, when TMPRSS2 now is expressed in these same cells, you can see that the cathepsins are much less important, and that TMPRSS2 is the preferred protease. However, in combination, both the inhibiting cathepsins and TMPRSS2 are the most effective in preventing virus entry. Okay, so just to recap, entry of the virus requires binding of S to its receptor, and then a proteolytic cleavage event that then allows for fusion of the viral membrane with the host cell membrane to release the genome into the cytosol. And then once the RNA is delivered into the cytosol, replication starts to happen. And the very first step for positive sense RNA viruses is always translation, because their viral RNA-dependent RNA polymerase needs to be expressed first. So in this case, ORF1A and 1AB are being translated, proteolytically processed, and then the individual non-structural proteins then assembled to form this replicase complex. And we'll look at that more specifically in the next slide. But what the replicase does is it makes more RNA genomes. And so if you focus on the right side, you can see that RNA replication happens in membranous vesicles, and I'll show a few images of that in a bit. But within those vesicles, the positive sense RNA genome is the template for the replicase to make a negative sense genome. This then results in this double-stranded RNA replicative intermediate, and then the negative sense genome again is the template for more positive sense genomes, which can be then packaged to make new virions. And so the replicase obviously is a very important protein, and is actually, even among the RNA viruses, a very special protein in case of the coronaviruses. And the reason is that typically, RNA-dependent RNA polymerases have low fidelity, so that means they make lots of mistakes, and that limits the size of RNA viruses to below 15 kilobases. However, I told you that coronaviruses have genomes that are around 30 kilobases in size, and so the only reason that these viruses can maintain viral fitness is that they have a replicase of higher fidelity, so one that makes fewer mistakes. And this is mediated by the exonuclease activity of NSP14. And so NSP14 teams up with the RNA-dependent RNA polymerases, so if it makes mistakes, those can be corrected. And shown on the bottom here is this nice cartoon of what the replicase complex looks like. And it includes several of the other non-structural proteins, which actually form a complex that has similarities with the DNA-dependent polymerases. And with for example, NSP9 is a single-stranded nucleic acid binding protein, and then NSP7 and 8 might form something that looks like a sliding clamp. But to show you how important this exon nuclease activity is, I want to just show this one piece of data from Mark Dennison's lab, where they took, in this case, SARS-1 wild type and compared it to a mutant that had a mutated exon activity. And what you can see, what they did is they infected cells, waited for one replicative cycle, and then sequenced all of the genomes. And then the mutations were aligned along the viral genome on the x-axis, and the frequency of the mutation in the population on the y-axis. And what you can see, just focusing on the black bars, the wild-type SARS makes few mistakes, but the XON mutant virus makes many mistakes in many different parts of the genome. And as you might imagine, viruses that have many mutations are very attenuated in vitro as well as in vivo. And so targeting exon activity is one of the antiviral strategies that is being employed to develop antivirals. Okay, and so just to show you these replication complexes, which are so important for the virus, we can see images here from SARS-1, and in general, all positive sense RNA viruses hijack intracellular membranes during replication, and you can see here what an infected cell cytoplasm looks like. On the bottom here, there's the nucleus. M is one mitochondria, and then lots of these membrane blobs. And when those are reconstructed in 3D, you can see these double membrane vesicles that are being made by the virus. And it's within these vesicles that the double-stranded RNA is found, and that's when new genomes are being replicated. And one thought is why these viruses need to rearrange membranes so dramatically is that it helps them concentrate the enzymes and substrates in a defined location, but also it hides away the double-stranded RNA, which I'm sure you're aware of is a very strong molecular pattern that is recognized by cells as a danger signal. And so these membranes are thought to help hide those patterns. And on the right, you can just see the as a time series, as time progresses, there are more and more membrane rearrangements, starting with rough ER and convoluted membranes, these double membrane vesicles, all the way to assembly. And so that brings us to the last step in the life cycle, which is assembly and release. And so once the proteins have been translated, the structural proteins S, M, and E are inserted into the ER membrane. And it is from there that they then move into the Golgi, and it's in the intermediate Golgi compartment that M is the catalyst for the assembly. So M will recruit both S and E, and then also N, which brings the genome along. And then the virus buds into the Golgi compartment to form the mature virions that can then be released by exocytosis. And one aspect of this is that not all of the S protein is being recruited into these new virions. And so when free S protein reaches the cell surface, and it's cleaved by host proteases, it can mediate fusion. And so you can have what's called the syncytia, and this is an image from an SARS-2 infected macaque. And so infected cells that are expressing S on their in the cell membrane can then fuse with uninfected cells to make these multinucleated structures, and this also helps the virus hide from antibody neutralization, because now it can infect a new cell without even leaving the protection of the cell membrane. And so after having talked about the structural proteins as well as the non-structural proteins that make the replicase complex, I just want to and by briefly talking about the accessory proteins. This is a summary of a few of them and their function in SARS-1. As you can see here, they are involved in modulating the immune responses, whether it's upregulating cytokines and chemokines or suppressing the induction of type 1 interferons and interferon signaling. And this was highlighted in a study in 2016, how important the type 1 interferon response was for disease symptoms development. And so this was Stan Pullman's lab that infected mice with SARS-1, and they could show that depending on the timing of the type 1 interferon response and the magnitude of it, that then affected the magnitude of the disease as well. If the type 1 interferon response was happened earlier or was reduced in magnitude, they also saw less disease. And so that brings me to the end of this brief excursion into the coronavirus life cycle. And before I turn things over to Dan, I just want to thank the people in my lab. They're an awesome group, and I look forward to the day that we can all assemble again. So this was our last lab picture before quarantine, and so hopefully we can resume those meetings